Welcome back to my channel Back and By World. In today's video, I'll be discussing the differences between eukaryotic and prokaryotic DNA replication. By DNA replication, it means that the DNA is replicated so that the cell can undergo, we can say, mitosis or meiosis and to generate the progeny, or we can say the daughter cells. So I'll be discussing the differences, the major differences between the prokaryote and eukaryote DNA replication, and let's begin with it. And first is the process. What kind of process, or when it happens, which phase it happens, at which stage it happens. So in prokaryotes, the DNA replication is a continuous process. There is a no specific phase at which we can note down the DNA replication. Whereas in case of the prokaryotes, it usually, or we can say, it occurs in the S phase of the cell cycle. Okay, usually the cell cycle is interphase G1, S, G2 and M phase, and the DNA replication takes place in the S phase of the cell cycle. Okay, and the DNA is the DNA which is present in case of the prokaryotes is usually circular in nature. Okay, they are usually double-stranded circular. Okay, whereas in case of the eukaryotes, it is a linear form, linear and double-stranded. Okay, and what is the site of the replication? What is the site of the DNA replication? We all know that prokaryotes is not having an intact, we can say, the nucleus. Okay, the DNA is not confined to a particular space which is termed as a nucleus, surrounding by nuclear membrane. And if we keep in that view of this thing, prokaryotes DNA replication site is cytoplasm itself. Okay, it occurs in the cytoplasm, whereas we know eukaryotes, they're having a intact nucleus, and the DNA replication, our genetic material is in the nucleus, and the replication of it takes place in the nucleus itself. Okay, so as to begin DNA replication, okay, we are having a double-stranded DNA. So like we are having a double-stranded DNA like this. It's like it's in the form of like this, right? So in order to start DNA replication, there should be a specific site. Okay, let me change the color. There should be a specific site. There should be an origin, or we can say the origin of replication. Usually the prokaryotic nuclear genetic material is usually smaller in size. Okay, so the origin of replication is usually 1 in number. Okay, we do not require much origin of replications because as the genetic material is smaller in size, and but it is in eukaryotes, the genetic material is very large. Okay, so as to complete that replication of the DNA replication in a particular time, okay, there is a time of S phase. Okay, so in order to complete the DNA replication in that particular time, number of regions are required. So the region of replication in case of the eukaryotes are multiple in number. Okay, enzymes which are involved. Enzymes which are involved in the prokaryotic DNA replication is DNA polymerase 1 and 3rd. Okay, whereas in case of the eukaryotes, it is DNA polymerase alpha, delta and epsilon. Okay, the Okazaki fragments, we all know that DNA replication occurs simultaneously. DNA replication occurs of both the strands. Okay, uh, of both the strands. One strand is usually synthesized in the continuous manner, and the other strand is usually is synthesized in the discontinuous manner, and the fragments are termed as Okazaki fragments. Okay, usually in the prokaryotes, the Okazaki fragments are relatively smaller in length in comparison to that of the eukaryotes. Okay, what is a rate? What is the rate? That is, rate means that how many number of bases are added per second. In case of the prokaryotes, the rate is very rapid. Okay, approximately I can say 2000 bases per second. Whereas in case of the eukaryotes, the process is relatively slower. That is, it is very slower, I think, in comparison to the prokaryotes, judging by the number. That is, it is usually 100 bases per second. Okay, and what are the results? What do we get after replication? In case of the prokaryotes, I'll get two circular chromosomes. Okay, two circular chromosomes, or we can say two circular chromosomes are obtained. Whereas in case of the eukaryotes, they are usually two sister chromatids. Okay, two sister chromatids. Like we can say the two separate chromosomes from one parent cell I get, that is the DNA replication, double stranded circular DNA. I'll get two circular chromosomes, whereas in case of the chromatids, that is, this is a single chromatid. After replication, what happens? It is having a sister chromatid like this and joined by the centrosome, centromere, sorry, centromere. Okay, sister chromatids are obtained. DNA gyrases, they are usually required in case of the prokaryotes only. They are usually not required in case of the eukaryotes. That was all about the differences of the DNA replication in the prokaryotes and the eukaryotes. Thank you so much for watching my channel. Please like, subscribe and share. Thank you.